So far in this workshop, we've looked at the mechanics of fitting linear mixed effects models, how to interpret the fixed and the random effects, and also discussed some different strategies for model selection for identifying the most parsimonious model. The final stage of the modelling process is model diagnostics, which is where we attempt to validate the assumptions that the model makes. This is important for ensuring not only that our model has a good fit and good predictive performance, but also that it's robust and doesn't violate any important assumptions that we make. On this slide, I've listed out some of the more important assumptions made by linear mixed effects models. Many of these assumptions are common to other regression modelling techniques that you may have used in the past. The first one is that we assume that the predictor variables are related to the response in a linear fashion. All of the modelled relationships follow a simple straight line, and we're assuming that that straight line relationship is an adequate way of describing how the response and the predictor are related to each other. As we discussed earlier on in the workshop, there are other techniques which allow for more complex model fitting and for those relationships to be described by curves rather than straight lines. But for a straightforward linear mixed effects model, we do assume linearity. The second assumption is that residuals have a constant variance. That is, the amount of scatter that's in the data that's left unexplained by the model is the same regardless of whether the response is taking low or high values. The third assumption is that the residuals are normally distributed. Throughout this workshop, we've been focusing on linear mixed effects models with a normal or Gaussian error distribution. We discussed earlier that if we are dealing with other types of data, for example, presence-absence data or count data, that different alternative error distributions may be more important. But certainly for many commonly used biological metrics, including many of the macroinvertebrate metrics, a normally distributed model is perfectly adequate. The fourth and the fifth assumptions here I put in italics because they tend to be slightly less important for these kinds of models. The models are either more robust to these assumptions, or just due to the nature of the data, these assumptions are less likely to be violated. The fourth assumption is that not only are the residuals normally distributed, but the random effects are also. And the fifth assumption is that the errors are independent. That is, there's no spatial or temporal autocorrelation. Focusing on the first three of those assumptions, we're now going to look at a function in the HE toolkit which allows us to quickly validate and test those assumptions. The function is specifically designed to generate a variety of diagnostic plots for a linear mixed effects model, that is an LMER model. You can see from the code on the slide here that to run this function, we feed it the name of the model that we fitted to the data. This might be the final model that emerges from our model selection process. We also give it the calibration data set we've used to develop the model. And we can optionally include a faceting variable if we want to explore not only how these assumptions are met by the model as a whole, but also how well the assumptions are met for the data from each of our individual sampling locations. This produces an output, which is a list of plots, so we can access each of those plots in turn using the double square brackets to extract the first, second, third, fourth, and fifth plot. On the next few slides, I'd just like to go through each of those plots one at a time and talk in a little bit more detail about what we're expecting to see and how we can validate these assumptions. The first plot is a scatter plot of the observed versus the fitted values. So the actual measured values in our data set are shown on the horizontal axis at the bottom, and the fitted values predicted by our model are shown on the vertical axis. The black dotted line is a one-to-one relationship, so a perfect model would be one in which the fitted values match perfectly to the observed values and all the black points fall on that diagonal line. That obviously doesn't happen in reality, and so instead we can draw a blue diagonal line through the data to show on average what the fitted values are and how those correspond to the observed values in our data set. This scatter plot provides a visual guide to the strength of the model fit. The more tightly clustered the points are around the blue line, the closer the fitted values are to the observed values. You'll also notice that the blue line doesn't follow the perfect one-to-one relationship. In any regression model, it's very common for the fit to be poorer at the extremes. The models tend to be quite good at explaining or being able to predict the values in the sort of the middle part of our data set, but for very high or very low values, those are inevitably slightly more difficult to predict, partly because they're less common and partly because they can often be subject to very specific localized influences which can be difficult to capture within this kind of model. Specifically, you can see that the model has a tendency to under-predict the high values and to over-predict or overestimate the low values. Again, that's a very common tendency in these models, and it just is just a slight warning or flag that using any model to try and predict the response under extreme conditions can be challenging, and we should all be wary about the quality of those predictions. This plot shows all of the observations in our data set. However, if we were to facet this by site, then we would get an equivalent plot for each of the individual sampling locations in our data set. And that provides a nice, simple and visual guide to the performance of a model for each individual site, and can help us to identify any sites where the model is performing well or any sites where the model is performing less well, where there's a weaker correspondence between observed and fitted values. The next plot helps us to assess the assumption of normally distributed residuals. It produces what's called a normal probability plot. Again, each of the individual observations in our dataset is represented by a black point, and they are distributed in such a way that if the residuals follow perfect normal distribution, those black points will fall along a straight diagonal line, which you can see on the plot. Outliers show up as isolated points which fall a long way away from the other observations in our dataset. So you can see, for example, on the slide, there's two observations down in the bottom left-hand corner, which could be referred to as potential outliers. They buck the general trend and don't follow the standard normal distribution. In this example, our dataset generally conforms to the assumption of normality. Most of the points fall along that straight diagonal line, with only small deviations at the upper and lower limits. Linear mixed effects models are relatively robust to deviations from normality, and we should only be concerned if the normal probability plot shows, instead of a roughly straight line, something more akin to an S-shaped curve or something that looks a bit like a skateboard ramp. If you do have concerns about the normality of the data, then you can either try transforming the response variable to make it more normally distributed, or trying an alternative error distribution altogether. If you think that, for example, your data are counts or proportions, then that might be a better strategy than trying to force the data to be normal. The third plot plots the residuals against the fitted values. So the fitted values are along the bottom, with low values on the left and high values on the right, and the residuals are on the vertical axis. This tests the assumption of homogeneous variances. Ideally, we would expect to see the same range of vertical variation or scatter in the residuals, regardless of what value the response takes. In this particular example, the data fulfills that assumption quite nicely. We can see, regardless of where we are on that plot, on the left-hand side or the right-hand side, we have a similar amount of residual variance, a similar amount of vertical scatter. Sometimes that's not the case, and it's quite common to see the degree of variation increasing with increasing fitted values. that results in a plot which has a funnel-shaped distribution of points with a very low degree of scatter on the left and then a progressively greater degree of scatter as you move to the right-hand side of the plot. That again can be an indication that a normal distribution is not the right error distribution for our data. Again, we can either try transforming the response variable, something like a log transformation may resolve the issue, or we may need to investigate an alternative such as a Poisson or negative binomial distribution. Fourth plot that we get from the function produces a histogram of residuals. This is really an alternative to plot 2 for assessing the assumption of normality. The red shaded area shows the overall distribution of the residuals from our model, and the blue line shows a perfect normal distribution which provides a reference point for assessing how close our residuals are to a true normal distribution. In this case, we can see that there's a very, very close correspondence between the distribution of our residuals and a normal distribution. This gives us a high degree of confidence that this assumption has been met. This plot is based upon the data for all of the sites in our dataset. However, if we were to facet it by biology site, as shown in the code on the right, then instead of producing that probability plot, it produces a series of box plots, which show for each individual site in our dataset the distribution of residual values. This can again be very useful for identifying any individual points which are outliers. So focusing on the black points which fall outside the normal range of residual values at each site, we can identify points which may be showing very atypical behaviour. And it also allows us to identify sites which might have an unusually high degree of variance. Lastly, plot 5 plots the residuals against each of the fixed predictor variables in the model. In this example, we have a model with two predictor variables, a Q10 variable representing high flow influences and a Q95 variable representing low flow influences. The residuals are shown by the green circles on the plot, and you'll notice that there's also two lines plotted through those points. The red line should be horizontal. If it's not, then it indicates that the linear effect described by the model may be an over or underestimate of the true effect. So if we see a diagonal red line, that's an indication that the strength of the effect of that predictor variable has been over or underestimated. The blue line is a smoothed line through the points. If this deviates from a horizontal line, then this indicates that the effect of the predictor on the response, as modelled, may not be linear, as assumed. In the Q95 plot on the right, you can see that the blue line is very close to horizontal and deviates only very slightly. That provides strong confirmatory evidence that our assumption of linearity holds true for that variable. For the Q10 variable on the left, overall it's not doing a bad job. The blue line is quite close to horizontal for most of the data set, but it does tend to have a tendency to dip down on both the right hand and left hand side of the plot. On the right hand side, we can see that the line is being pulled down by just 3 points which have an unusually high Q10 variable. So those points do have a relatively high leverage or influence on the model, but there's only 3 points, so we probably wouldn't be too concerned about that. A potentially greater concern is the tendency on the left hand side of the plot here, which shows that the model tends to overestimate the response when the Q10 value takes low values. There's actually a reasonable amount of data here to support the idea that we may not be looking at a perfectly linear relationship. Depending on how important or influential we think this might be on the model and the predictions we want to make from the model, we might be warranted either in including a squared term to create a quadratic model, or we may wish to use some of the more flexible models that we covered in the earlier tutorials, which provide a way of describing these relationships in a non-linear fashion. So together, this diagnostic function provides a series of visual plots which allow us to validate the assumptions of our model. And validating these assumptions is important to make sure that we have true confidence in the model and it's a representation of our data. If you would like to explore the code that we've covered in these last few tutorials for model fitting, model selection and model diagnostics, then please hop over to the POSIT Cloud project that we've set up to accompany this workshop. There you will find an R script called Practical Exercise 3, which contains an example dataset and some example code that you can run to illustrate all the different approaches that we've covered.